Hello, everyone. I see that we are trickling in. We're going to go ahead and get started because this is a tight session. So this is our second keynote on registries and GRI disorder phenotypes. Today, we're very excited to be joined by Dr. Alan Byatt, Dr. Wendy Chung, and Dr. Tim Benke. We're going to kick things off with Dr. Wendy Chung, who's the Chief of Pediatrics at Boston Children's Hospital and is closely involved in working with Simon Searchlight, who's going to talk to us about GRI disorders with Simon Searchlight.
Thanks, Megan. I'm just going to jump right into it, and hopefully this is presenting back your information to you and giving you some sense of what to expect going forward. So Simon Searchlight, we've been around for over a decade and we're going strong. Just to say that I call them the GRI conditions, are a perfect paradigm for how to be able to do comparisons across conditions, and you're powered by a very large team. Both it's Safari or Simon's Foundation Autism Research Initiative, as well as my team at Boston Children's and a team around the world. As we're doing this, we're here for the long haul. We've been here for 14 years and going, and we continue gathering data and making it de-identified but available to the world of researchers free so that they can use both your information and biospecimens, and it hopefully has less burden on you, giving this information back to you and also your providers, and hopefully being able to think ahead about what we're going to need for clinical trial readiness and to be able to get to therapies. And so as we're doing this, we're trying to make this, as I said, as easy on you and de-risk it and bring more researchers to help you to achieve your goals. As we do this, I'll say we have both initiatives that I lead and Spark, Simon's Foundation Powering Autism Research for Knowledge, for people to get a GRI diagnosis. And so we have many of those participants in the United States coming in to join you. It doesn't happen very often, I'm glad to say, but we do have an initiative to be able to look at brain pathology for individuals in our community who pass away. And then, as I said, we distribute all this information through Safari. That's really for the researchers out there. In doing this, as I said, Autism BrainNet, the main thing I'm just going to highlight is this is phone number down on the right, and the URL is available 24/7 should it be ever be necessary. I'm highlighting here the GRI genes that we have within Simon Searchlight, and we're continuing to expand this as more genes are added. And so you can see on the right more genes that are being added soon. We have the dynamic ability to add genes as well as add languages around the world. In doing this, we're supporting individuals for online research to make it available to as many people as possible. And here I've listed all the different surveys that are done. The column for baseline means when someone enrolls, and annually means that once a year we follow up to understand how people are doing over the life course. And as you'll see, we understand much more about how people are doing in the first parts of their life up until the age of 20, and much less information longer term. That data gap is something we're still very actively trying to fill so that we know how you can help to navigate the person in your family going forward. The way we've done this online, it allows the person to, I call it choose your own adventure, in the sense that you can do this no matter what time zone you're in. You can start and stop and come back to it later. You can have a genetic counselor to speak with you, both in terms of learning more as well as having them maybe explain questions that aren't obvious or where you have complicated answers. And then we have the genetic counselors going through your answers to make sure that they make sense, and then clarifying anything with you. So we hopefully have something for everyone in terms of being able to do this in a way that's easiest for you. We have a dashboard to make it a little bit easier to know what your next step is. And so if you log into your dashboard, you can quickly pop up and see the next thing that you need to do. But just know that this is, as I said, ongoing. And so understanding what's happening over time is incredibly important to researchers. I'll also say that we realize it takes your time and energy to do this, since we do have a small token of appreciation with an Amazon gift card that can be used for individuals mostly around the world. We tried to get something that was somewhat universal with that. I'll also say that we're listening to the community and continuing to evolve what we get. We'd like to hear if there are any researchers listening to this about things that we should be adding that'll be helpful as we understand the conditions better. We are trying to right-size the assessments to the ability of the individuals in the community. So we're adding the ORCA, some additional behavioral measures, doing increasingly, in many cases, in-person measures even for individuals who are partnered, for instance, instance, if they're having a family meeting, in some cases to do things in person, then collecting certain medical records, and especially for this community, EEG data or electroencephalograms. In doing this, we have reports that come back to the community to be able to understand, hopefully in a relatively easy way, what we're seeing. So with this, we have these updates that we send out, and they reflect similar information to what I'm talking about today. We have individuals reports for you personally. So if you complete some of these measures online, some of the things that are related to the brain and behavior, you get back your information personally. But then you can also see these summary snapshots so that you can see where you fit in with the rest of the community that are gene specific. So this one, for instance, is displaying for GRIN2B, similar to what I'm going to show for the data later today. And as I said, help you understand whether you're typical or whether you might have things that might be special, because everyone is special, but we're special in our own unique way. In doing this, as I said, I want to reassure you that your information is protected. It's de-identified, so no one should be able to figure out or find you. But on the other hand, you're represented. And as I'm going to show you, it's incredibly important to be represented because everyone is so unique. I'm very pleased to say that in terms of how many people are using the data, we have over 800 researchers around the world who are using not just the GRIN data, but across Simon Searchlight. And so with this, it's very actively used. Those researchers in universities around the world, also in companies developing treatment. So with this, very heavily utilized. And again, this helps you to leverage your investment so that more people are working for you. With this, we have some people have donated not only their information, but also a blood sample. This can be done through, especially in the United States, but increasingly around the world as well. And I won't have time to go into the details today, but we do have it set up in the United States where you can go to a local Quest lab, or in certain cases when we have one time in the year, a time to be able to come to a site where we have a phlebotomist drawing blood. And in part, this is used to make induced pluripotential stem cells. That's a very significant investment at the Simons Foundation to be able to support that. And those cells can be used by researchers around the world to be able to make brain cells in a dish, organoids, other things to be able to understand how someone might respond, for instance, to a therapy or something they're developing. This whole process of modeling is important, as I'll show you, because everyone is unique and there are various different genetic variants, not just because there are different genes, but different variants in the gene. And we don't know that all of those variants will behave exactly the same way. There might be some nuanced differences, and they might require some different therapies. And so it's important as much as possible to be represented both in terms of data as well as in terms of specimens for the models that researchers are using. We also have, and this is mostly for the research community, the ability to connect you directly to the GRIN families through something called Research Match. So if you have some questions, some data that you would like that we don't already have covered, you can directly work with us. This is a free service, I want to emphasize, so that we can help you administer this information to the participants and be able to get that information back and connected to the other information that we've been collecting on families. So this hopefully takes the burden off of you, and especially if you're not someone who's tied into the research community, the patient community, this should make it much easier for you. So with that introduction, let me just dive into the data in terms of the GRIN registries. I'm going to be talking first about GRIN2B. This is actually this condition for which we have the most data, which is why I'm focusing on this. I don't mean to leave anyone out though, so I'll try and give a little bit of information about some of the other genes as well. But just to show you, we've had 157 individuals registered at the left of the slide. As they go through, we verify their genetic diagnosis by reviewing their genetic test report, and then they begin sharing their medical information and other surveys, and in some cases provide a blood sample. And then, as I said, get updates every year. So not everyone has done everything, but it's a great group that has, for the most part, done most of the things in the steps along the journey. This is what I call a lollipop plot, because these are the lollipops. And this is the GRIN2B gene shown in a stick figure form for the researchers, indicating the different domains here. And each one of these red dots here is an individual participant, a unique participant. And I've shown the different genetic variants in the GRIN2B gene. The numbers afterward indicate that there's more than one person with the same genetic variant. These are unrelated individuals, almost in all cases. So these have occurred in different families. These individuals are not related. And so those may have special significance. But what I want to emphasize is how much variability there is. There are many different genetic variants, and that becomes incredibly important to researchers as they're trying to predict for you what to expect, what treatments might be most effective, and long-term what treatments, as they develop treatments, to make sure no one is left behind. Because of this, as I said, it's incredibly important for you to be represented as much as you can be. And so an easy way to do that, as I said, through Simon Searchlight, because so many people, so many researchers are coming to Searchlight to access the data. Within this, as I said, we have already 11 induced pluripotential stem cells. So for any researchers who might want to use those, as well as in many cases, samples that could be made into induced pluripotential stem cells or used for other reasons, depending on what researchers might want to do. So a resource in a library that continues to grow. This is for the research community. For the most part, these are the specific alleles that are represented within the iPSCs. You'll see a combination of missense variants as well as some truncating variants here. All but one of them is available already. And again, if you go to Safari Base, you can request those and they're basically free. All you need to do is pay for the shipping fee. Within this, we also have individuals not only for GRIN2B, but I'm showing here on the left 3 other genes, GRIN1, GRIN2A, GRIN2D. As you can see, these communities are a bit smaller. The frequency of the conditions in the general population is less, and that's why the communities in Searchlight are smaller. You can see the numbers coming along nicely. And for researchers who may want to do comparisons across the genes, again, all of this is available through the same place. I'm just going to very briefly for the researchers show similar stick figures of the genes to show that, again, this is very typical that we see largely missense variants. We see largely unique variants, although some are recurrent that we see over and over again. And again, making the point of how important it is for individuals to stand up and be counted to make sure that they're not left behind. For GRIN2A, again, there's a theme, and it's important that there is a theme because many researchers are working on this theme. Again, the types of genetic variants, very similar in terms of where they are in the gene, also similar in terms of the diversity of the variants that we're seeing, and many of the biophysical, I think, effects are going to be similar across the family as well. And then finally for GRIN2D, again, a smaller community so far, but again, a clustering of those variants that we see, and not surprising, in fact, that we see this clustering. Across these genes, this is now the GRIN1, GRIN2A, and GRIN2D. Again, similarly, although it's still evolving, a library of specimens that are available for researchers to be able to use. So finally, in the last few minutes, I'm going to go over now some of the clinical data for GRIN2B. I apologize, I've just got time to do GRIN2B, but the themes are very similar across the GRIs. The point that I'm making here is this is a distribution by age of individuals who I'm representing. There are 93 total. You'll see that most individuals, as I alluded to before, are before the age of 20, and in fact, most of them still before the age of 10. It's not to say that there are not adults and older individuals out there. They simply are not yet accessing genetic testing to understand their diagnosis from a genetic point of view. So hopefully, as it becomes more important to know your genetic diagnosis for a treatment, then hopefully more people will get access and in fact perform genetic testing. The good news for those of you who have heard me speak before and know from your doctors is that there are not a lot of surprises. So if you want to take away one thing, this continues to be a condition related to the brain and behavior, but there are not a lot of surprises in terms of other complications, either cancer or long-term other issues that are coming up that are unexpected. And this does not look so far like it's a neurodegenerative condition, mostly a neurodevelopmental condition. Pretty consistently, everyone has concerns in terms of cognitive issues that lead to delays in terms of achieving certain milestones. About a third of individuals have autism, but there are also, as you'll see, a lot of other issues with attention and to a certain extent issues with anxiety as well. Related to that, I think it's related to the brain's involvement in the rest of the body, but we see issues with muscle tone, issues with coordination, sort of how smooth movements are, being able to be adept in terms of both gross motor or moving up and down stairs or walking or running, but also fine motor or hand movement skills as well. There can be other issues, and there have been some misdiagnoses, I think, in terms of things like cerebral palsy, where really I think mostly it's a descriptive term in this case, but really due to the underlying GRIN2B diagnosis. Of course, epilepsy is a common concern in about a quarter of individuals. It's many different types of epilepsy. I think one of the things we are still interested in understanding is whether or not this is mostly something that we see this largely within the first few years, and I think we know what we're dealing with. Again, I won't say that we're seeing many surprises yet, but we're still watching in terms of adulthood to make sure that we don't see surprises there. As I said, for the most part, individuals are having seizures early. So most individuals, if you've got individuals who are 5 years or older, it's most likely that you've seen the first seizure if you are going to be in that quarter of individuals who have seen seizures. For as they've been doing it, many individuals have needed medication for their seizures. And as people have grown up, as I'll show you, there hasn't been a clear winner in terms of seizure medication that has been sort of a cure-all for everyone. In going forward, the other medical problems have largely been, I think, related to issues that are secondary to the brain. So some of the gastrointestinal issues, constipation, diarrhea, even alternating between the two, I think related to the innervation or the nerve cells that go to the brain, to the gut rather. Other issues related to vision, some issues related to orthopedic issues of scoliosis, ultimately, I think in part related to the brain issues, and for certain individuals who have needed help with feeding and needed a gastrostomy tube, again, that related underlying to brain issues. So for the most part, I think this is really, you should think of it as a brain condition. You'll see on the upper part here, there are a few individuals with other sort of issues, but this is really very frequent to what we see within the community of individuals who have either just the general pediatric population or to a certain extent the population of individuals with neurodevelopmental disorders, and certainly no surprises. As I said, most of the surgeries have been related to gastrostomy tubes, but occasionally other, especially orthopedic surgeries. And as I said, from the orthopedic point of view, largely scoliosis, some problems in terms of either with the hips or sometimes the feet, but largely scoliosis. The endocrine issues have been largely around growth and either gaining weight, sometimes requiring the gastrostomy tube, or in some cases being a little bit shorter. And the vision issues are important for you to continue seeing your ophthalmologist or the eye doctor. There can be issues that are easily corrected with glasses, but in some cases that need surgical correction. For instance, if there's a lazy eye or cross eye. For medication, medications have been used for a variety of these symptoms. Again, many things that are relatively straightforward medications for gastrointestinal issues. Sleep issues have been an issue for some. And then also, of course, anti-epileptics or for behavioral issues, antidepressants. Within this, we'll see as this changes over time, and in particular, the ones that are related to behavioral conditions as those change as we go through adolescence and young adults. Um, as I thought I would say, in terms of medications that are most effective, we always ask that question because I know you're asking that question yourself. What we can see here is that for different individuals, different anti-epileptic medications have been useful. As you can see, there's no clear winner. Different people have found different medications to be useful going forward. And so do share this information with your neurologist if you're having trouble with breakthrough seizures or if you're new to the community. But again, in a good way, many people are treated, but there hasn't been a single medication only that's been effective. Many individuals ask, what can I expect going forward? And so we do something called a Vineland Adaptive Behavioral Scale. You actually fill out the questions and report what you're seeing as your child is developing. And each of these next couple slides are going to show individuals who are different ages on the x-axis here, and then how they're progressing. This is their functional age equivalent in the y-axis here. And you can see that individuals are steadily gaining skills over time. This graph is related to expressive language, and each of these dots is a different person. So in terms of being able to speak and expressive language, this is slow but steady and increasing over time. If we look at personal care skills and individuals' ability to dress or undress or bathe, things like that, again, nice and slow and steady, but increasing over time. And as we think about social skills, being able to make eye contact, having interaction with other people, having friends, with this again, increasing over time as we're making progress. And then gross motor development, or being able to, as I said, walk, run, skip, go up and down stairs, improving ability over time, and some individuals that are making significant gains as they're getting older. They also have some challenges from a behavioral point of view. This is for young children, less than 5 years of age. I'd say attention is especially an issue, and attention sometimes leads to wandering, which can be a concern. And this problem with attention continues for individuals age 6 to 18. They may also be distracted, hyperactive, need attention, and be fixated on certain things. So this is a somewhat typical profile that we see. So in summary, and I know this was a quick run through, what I hope you'll see is that there aren't surprises, there are continued concerns, but individuals are developing skills over time. And I think a lot of it is focusing our energy in the right way to support those individuals. And in the future, we'll be expanding, of course, as we have more data in terms of subdividing, looking comparison groups, GRIN1, GRIN2A, others. But as you're hearing from this meeting, investigators are extremely interested in understanding the nuanced differences between the genetic variants. And with these data available to them, I hope they'll make great strides with that. So I will pause here and continue on.
Thank you so much, Dr. Chung. So I do see that there are a couple of questions in the chat. We're going to try to hold those for the end. We still have a couple more presentations to get through. So we're going to go ahead and transition to Dr. Byatt now, if you want to go ahead and share your screen.
Sure. You should be able to see it now.
Yes, perfect. Thank you.
Amazing. So thank you for having me. This will be about the research we've been doing for GRI related disorders. And if you're interested, this paper has just recently been published in Brain. So let's start. This will be our stakeholder for today. So how do we deal with a small girl who has a GRI variant that is de novo, meaning that her parents do not have the same variant. So it's new in her. Do we say it's just something that's there? Is it disease causing? How do we deal with this? And the reason why this question is important is because GRI is X-linked. X-linked means it's located on the X chromosome, and I'll get back to why that is important. We have 4 GRIA genes, and these GRIA genes create different subunits that go together and create these, what we call heteromeres. So normally, most commonly, 2 GRIA2 and 2 GRIA3 subunits go together and create this AMPA receptor. And then there is an activating substance in the brain called glutamate that binds to it and then activates the receptor. This receptor is mostly expressed in the brain, and here you can see it highlighted in red. Let me put on my cursor there. So this is the receptor encoded by the GRIA genes, and here is the NMDA receptor that resembles this very much. And what we normally know or have considered is that because this is an X-linked disorder, we always consider males to be affected, while we normally think that a girl would be a healthy carrier because she has an extra healthy copy of the X chromosome. And this leads us back to this girl. So how do we deal with her should she has a mutation in GRI3 that her parents are not having? The problem is that there's very few papers, very few patients reported out there with GRI3 related disorder, and there's hardly any functional testing, meaning we as physicians have very limited knowledge and have a problem counseling patients like this. And this leads us to the current project. And this is the outline of the project. So we start with a patient that has had a variant in one of the 4 GRIA genes identified. This could be any gene, any variant that is either known to be disease-causing or a variant where the geneticists say this is of unknown significance. We do not yet know whether this is disease-causing or not. So these are the kind of patients where we are interested in. And we have a database that I'll come back to where we sort of interview the families, we put the clinical data in there, and then in the lab, we functionally test the human GRIA variants to find out whether this is a mutation that is affecting the function of the receptor, and if it is, whether it's a gain of function, so the receptor is overactive, or whether it's a loss of function, meaning that the receptor has lost its function. And in the end, we sort of do some drug trials and then counsel you as families or your doctors so that you can get the correct treatment at the correct time. And I just want to take one slide description of the functional testing that we're doing in the lab. So most of the experiments we're doing is based on a model that is well known and well described based on frog cells. So we harvest the cells from the frogs, and then we inject it with a copy of, let's say, GRIA3 that we want to study. And the sort of this, what we inject has the human mutation. These frog cells do not normally express the AMPA receptor. So when we sort of give them a copy of the receptor with the mutation, they start building the receptor on top of the cell surface, and then we can start injecting the oocytes and putting different chemicals on top of it and see what kind of current is going through this receptor. Is it a normal current? Does it look odd? Is it too active? Does it have reduced function or not? And I'll get back to some of the results that we can find. So for this project, we have so far found 40 different missense variants from GRIA3 patients worldwide. And this is one of the experiments that we are sort of doing. So here in the left, you can see that we have here an oocyte where this is what we call the wild type oocyte. This is how the oocyte will behave if we put into it a copy of GRIA3 that does not have any human mutations. So all the receptors here on the cell surface will be normal functioning. And you see that when we put increasing amounts of glutamate on top of it, you get a higher current here, and then eventually the current cannot get higher. We do the same with human mutations. So this is from a human being that had a mutation on position 609. And what you can see is the curves here looks very much alike the wild type. So in this case, we would normally go back to the families or the clinicians and say that with the knowledge we have here and with the sort of assay that we have, we think this is likely to be a benign variant. While for this patient, which could have been our stakeholder here, you can see that this is a gain of function variant. So even very small amounts of glutamate starts giving you a current. So the currents are much higher. So this is what we call gain of function. Whereas down here, this is another human mutation on position 776 on the protein. And here you can see there's hardly any current coming. So this would be what we call loss of function. So this is how we sort of use these cells to give us an insight of whether this is doing something or not, these human mutations. This is an overview of the AMPA receptor, and this is a 3D model. The AMPA receptor here has what we call the upper part of it. This is the part that communicates with the outside surface. Then there is what we call the ligand binding domain. This is where the glitamate binds and activates the receptor. And then there is this part here that binds to the cell surface. We call it the transmembrane domain. And each part here has been sort of blown up. And you can see spheres here that are representing a human mutation. And the gray spheres are the ones that we so far have not found to do anything to damage the receptor. So these are the ones that we will call benign. And the green and the red ones are the ones that are damaging. So the green ones would be the gain of function variants, and the red ones would be the loss of function. You can see what we can see so far is that it's mostly variants from position 400 and above that seem to be disease causing so far. If we look at the variants here based on the green and the red spheres, we have in total 25 patients, meaning 11 females in total. And most of them are children, so with a median age of around 9 years, but there were some that were as young as 5 months, and there were also some that were in their 40s. So there is this gives us sort of an insight into how the disease develops over time, although most of them were children. What we can say, if we merge all the gain of function and the loss of function variants, we can say that all of the patients did have intellectual disability or developmental delay of some sort. But there is a range. It ranges from something that is mild to something that is very severe. And this is the mild spectrum. So the mild spectrum, the kids have very normal early milestones, but then early in infancy, we start seeing that there's problem with motor development, with speech and social problems. The children start learning to walk, but the gait is unsteady. And if they have epilepsy, it is relatively easily treatable. And this is the worst part of it that we know so far. Here, the children are affected at birth. They have very abnormal body tones that they can either be very flaccid, or they can be extremely stiff, which is the case of this girl. They have feeding tubes or feeding problems. They have no or limited eye contact, hardly reach any of their milestones, and the epilepsy is devastating in most patients. So now the question comes is, can we clinically see any difference between patients that have gain of function or loss of function? I'm going to show you some videos, and I want to thank the clinicians and the families for donating this. Here we have, once again, our stakeholder and another girl, and they both have a gain of function. This girl has a strong gain of function. This one has a mild gain of function. And you can see this is a small baby with a feeding tube. She's very stiff, and even mild touch or sounds makes her twitch, which is quite uncommon for a small baby. Later on, the clinician starts taking her up, and as you can see, this poor girl is extremely stiff in her body and has really difficulty relaxing. Looking at this young girl, we can also see that she's also quite stiff, has the problem sort of relaxing in her body. She has a twisting of her left arm, but she is actually able to sort of interact, look at the physician.
Metti giù la testa, dai, giù tranquilla. Oh, Brava. La mamma viene da questa parte, così almeno ti vedi.
So these were the gain of function girls that I had some videos on. In comparison, we have here two boys that have a loss of function. And you can see this young gentleman in his wheelchair. And you can see how so that he stands out as being very weak in his muscles. The mouth is open, the eyes are hanging. So he sort of has problems, even struggling to sort of move his arms and legs against the gravity. And he has a strong gain of function. But of course, not all boys are like that. Here, a young man also with a loss of function mutation. You can see he is clearly able to stand and walk, and he can interact. But of course, as you can see, despite that I guess he is about 5 or 6, that he does have language problems. But let me show you the video again. You can look at his standing and his body tone. It does seem a little bit wobbly and low in this young gentleman as well. So, sorry. If we look at the currents, so this is a picture of sort of what kind of current you would expect to see in the synapse. So this is the wild type. Let me put on laser one more time here. So this would be sort of the signal you would get in a synapse, which is where one cell communicates with the neighboring cell. You would normally have a receptor being closed, and then it opens and closes again. While for the, sorry, once I'm on call, sorry. Jeg sidder i et møde, du bliver nødt til at ringe tilbage. Er det okay? Godt, hej, tak, hej. Sorry, multitasking. So what we see for most of the males is that they have inherited loss of function variants that basically give you a no current over here. While the females have these gain of function variants where you can see the receptor opens, but then it takes a while for it to close, or over here that it does not close at all as it should. So again, these are the signals we see for gain of function in females and the loss of function in males. And then when we start to merge the clinical data with the functional data, we start seeing some patterns. So up here is what we see for epilepsy, for gain of function compared to loss of function. What we see is you can have epilepsy in both groups, but if you have a gain of function mutation, you tend to start seizuring within the first month of life. Whereas if you have a loss of function mutation, the epilepsy often starts around 1.5 years of life. So should you have a variant that that has not yet been functionally tested, just looking at the age of onset of seizures might be sort of a clue to find out whether this could be a gain of function or loss of function variant. If we look at supporting symptoms, we can see that the stiffness and the startle phenomenon you saw in the small baby is something that is often linked to gain of function, while the reduced body tone and sometimes even sleep disturbances is a feature we see for loss of function. So take-home message is that both males and females can in fact be affected, and that the males often have variants inherited from healthy mothers, and these variants often cause loss of function, while the females have gain of function variants that are not inherited from their parents. And of course, that we can have clinical biomarkers that can help us to sort of predict whether a variant is gain of function or a loss of function while we are waiting for functional testing. Lastly, I want to let you know that we do have a database, just as the Salmons Foundation and Wendy Chung talked about. They're very beautiful and detailed database. We do have at the Danish Epilepsy Center a database that could sort of include all GRI patients, but is mainly designed for GRIA research, where we are sort of having physicians or you as families to put in clinical information about your patient, about the patients. We're also collecting EEGs and MRIs and videos so we can look carefully for movement disorders and body tone. And then there is, of course, and we'll tell you about this at a later time, but there is potentially precision therapy possible. We found out that perampanel is an excellent anti-seizure medication for most patients with epilepsy and gain of function mutations. Earlier today, we heard from Patty about her son with a loss of function GRIA3 variant, where she had tried off L-serine and has had promising effects for this. So L-serine could potentially be a treatment for loss of function variants, and this is also something that we're interested in. Lastly, I would like to thank CureGRIN, all the many collaborators worldwide, and Novo Nordisk Foundation for supporting this research, and my collaborators at the different university hospitals in Copenhagen. That was it. Thank you.
Thank you so much, Dr. Byatt. So if anybody has questions, if we can pop them in the chat. We've still got to get through Dr. Benke's talk. It looks like Dr. Chung addressed some questions. If we have any time at the end, hopefully we'll be able to get to that too. But right after this, we have another keynote, so it's going to be a busy afternoon. Jeff, if you could go ahead and play Dr. Benke's video.
Hello, everybody. Sorry I can't be there with you live. My name is Tim Benke. I'm at the University of Colorado School of Medicine and Children's Hospital, Colorado, and I'm going to talk to you about the Global Registry for GRINs. This is a partnership between a lot of players throughout the country and world. Both myself, Dr. Park at University of Colorado, Johannes Lemke, Ilona Cray, Chiara Klockner at Center for Rare Diseases and Institute of Human Genetics at the University of Leipzig, Steve Trinellis, Hongji Yuan, Scott Myers at Emory University, and Dennis Lau at the Cleveland Clinic. These are my disclosures, nothing really relevant in terms of conflicts of interest associated with this talk. So the hypothesis is that changes at the genetic level in GRI genes will result in changes in GRI function, which will inform different phenotypes and therefore treatments. And the phenotypes that we're talking about are different developmental milestones, regressions, epilepsy, storms, changes in MRIs and EEGs, vision, movement disorders, growth, puberty, medications, therapy, sleep, behavior, GI, autonomic, and even other illnesses and longevity. So where we are with the registry as of April of 2024, you can see the total numbers that we have in the registry. There's 335 individuals registered with GRIN1 changes, 360 with GRIN2A, 305 with GRIN2B, 46 GRIN2D, and on down the list with all of these different disorders, so that we have a total of over 1,000 individuals that have registered in with us. And I really appreciate everybody who's done this. So what does the result of our analyses look like? This is a snapshot of where we were in the recent past with, as you can see, smaller numbers than we currently have. And we're in the process of updating all of these numbers. But basically, the idea is with the different disorders, we want to characterize what does everybody look like in the database in terms of the different challenges that everybody has. Such as, for instance, you can look at GRIN1s. There's 187 individuals. The average age of people in the registries is 8.2 years. The oldest is 35 years. 100% of people have intellectual disability. 55% have an abnormal MRI. There's a large proportion with seizures, and this starts at a relatively early age. And we can see that this is different when you compare the different disorders, such that there are different proportions with seizures in the different disorders, such as 48% with GRIN 2B, and these ages of onset may be different. So where we are with these analyses and new initiatives, we are working on another GRIN 1 paper where we're analyzing the larger cohort. There's a GRIN 2A paper that's coming out soon. There's another GRIN 2B paper, again, with analyses of a larger cohort. There's a GRIN 2D paper in process. And one of the important things that we're noticing is that there are a number of individuals who have the same variant as other people. And so that's why it's really important that even if you find that your variant has already been analyzed, and we'll talk about what that means in just a minute, it's important that you go ahead and register with us, because we would like to know if different individuals with the same variant look similar or not. And this is really important to us as we try to go forward and understand what's going on with the different GRIN disorders. Another thing that's really exciting is that a Center of Excellence is open at Children's Hospital Colorado, run by Dr. Kristen Park. And there is another Center of Excellence that is in the process at the NIH with Catherine Roach and Sarah Inati. So one of the questions that often comes up is where is my functional analysis? Well, if you don't see it on one of the different websites that I'm about to show you, that basically you have to enroll in the registry in order for us to proceed with obtaining the functional analysis. So once that functional analysis is available, it can be provided to your physician at their request. They just have to email me at my email address here, and I will get back to them. And generally, this is working reasonably well. Providing this directly to families implies a clinical relationship. And really, what we have with you is a research relationship. I do often call families because it's easier than an email, but it's what's important to know is that I'm providing the information that we have on a research basis. So the other way that people can get their functional information, and primarily the main way that you will get this, is through the GRIN portal. And you can Google GRIN portal and it should come up, but this is also the web address here, and I'll show you how to navigate that. So what do you do with your functional analysis? So basically, what this functional analysis is saying, whether or not your particular variant is causing increased function of a GRIN receptor channel or loss of function. So there are a number of different assays that go into making this determination. It's a lengthy process. There's a team of scientists that are putting this together. And once those results are available, they're reviewed by Dr. Train-Ellis. And this is not clinically actionable, but it is necessary for clinical trial research and natural history studies. And two examples for this: in a natural history study, what we're thinking is that gain of functions may have different symptoms at different ages compared to loss of functions. What we're seeing in the previous slides that I showed you before, for instance, with regards to age of onset of epilepsy, is that that while an average is reported, those distributions are bimodal, so that different people it appears some are happening at early and another group is happening later. And what we would really like to know is this influenced by whether or not you're a particular gain of function or loss of function. And so this is important because future clinical trials will include only gain of functions or only loss of functions, or vice versa. And the reason why is that the drug used in these studies will be specific for one versus the other, and the wrong drug may make symptoms worse. So here's what the GRIN portal looks like. So when you go to the portal, basically you're going to see that there are a number of different options that I want to point out. One is, sorry, this arrow is supposed to flash such that you know to click on either variant analysis or clicking on GRIN registry for what you want to do. But let's talk about what happens when you click on variant analysis. So you're provided with the page, and what you do is you put in either the information on your variant at the protein level or the variant at the DNA level. And this information is obtained from your genetic report. And then you either click on search at the DNA level or you click on search at the protein level. And then what happens is it comes up with a new screen that says that some information about the variant. And one of the things that it comes up with is it says it's likely pathogenic. And then there's another button here where you can generate the report. When you generate the report, it says not only it's likely pathogenic, but it then gives you the functional consequence down here where it says likely loss of function. Now, there's different levels of determination. There's the strongest, which would be likely loss of function, and then right underneath that is possible loss of function. Then there's an area in the middle where it's indeterminate. And then there's possible gain of function and likely gain of function. So essentially in clinical trials, you will segregate and put into a group the likely or possible loss of functions or the likely or possible gain of functions when you're doing either segregation as part of the trial or when you're trying to understand better what your phenotypic information means in the registry. So another thing I want to point out here is this little box there next to it. It says this is the number of individuals with this particular variant in the GRIN registry. And this is what we're looking for, is we're trying to get as many people as possible with some of the more common variants, such that we can be more confident in saying that individuals who have a particular variant, they're more likely to have these particular challenges. And it makes our ability as clinicians, when we have all of this information, to provide better medical advice. So if you were to click on the registry button, you come to this page. And essentially, when you participate in the registry, it depends on where you live. If you live in Europe, Asia, Africa, or other, you click on this button here to participate, and you're sent to Johannes Lemke's portal. Or if you are in North, South America, or Australia, then you click on this button here, and you're directed to the registry that lives in Colorado. Both of our registries are linked, such that all of the data comes together into one global registry, such that we have the power of numbers as we go forward with this research. So when it comes to the registry, some of the questions that I often have for you folks are, why haven't you enrolled? And we also have the opportunity to provide updates, and we're really looking forward to people provide those yearly updates because we want to see how things are going for you. So the way you do this is you contact the registry, and I'll show you the links again on the next page. And then the families will email a copy of their full genetic report, all the pages, to the registry. Then myself or Dr. Park or Dr. Zlemke and Cray will then verify eligibility for enrollment. Our study coordinator will then email a link to a consent. You agree if you want to participate in the study, and then after that's completed, our study coordinator will verify this and then send you links to a questionnaire. And these are questionnaires that you fill out online, and they tell us a lot of different things about your family member. Then we notify CFIRV or Emory and Dr. Trainellis' group to proceed with functional analysis. And these functional analyses are happening and very pending upon available funding. And when they're completed, they're then uploaded to the GRIN portal. It's important to note that obtaining the functional analysis is a very lengthy process, and it does take several months to be completed. And it's important to note that we're not testing all variants, particularly we're not testing nonsense variants at this time because they're all presumed to be loss of function. This whole process, we only have one study coordinator, and to this point, we know that families are anxious, but it can often take 3 to 4 weeks for us to process your requests as they go through the system. So thanks for your patience. So the portal again is grin-portal.broadinstitute.org. And when you enroll in the registry, if you're in Europe, Asia, Africa, or other, you go through this email, grin@medicine.uni.leipzig.de, or North and South America and Australia. That's grinresearch@cunchus.edu. So one of the things I want to talk to you about, because this comes up a lot with regards to your functional analysis, is N-of-1 trials. And these sorts of approaches are individualized with outcomes for each person. And the outcome measures depend on a preference, because really you want to know if the drug that you're giving is part of this trial, if it's working. And you want to make sure that whatever change you're seeing is meaningful. And this, of course, is going to be subjective to each person's experience. But it's important to note that the FDA thinks that this sort of approach is very important with regards to meaningful change. And what the challenge is really to know how, if something is improved, whether that would be meaning to patients in general. And in a lot of these cases, that prior safety is not established, and safety measurements may be arbitrary, but it's important. You need to measure something and follow things in a reasonable fashion. And if things are getting worse or improved, you need to know, was this going to happen anyway, or was it as a result of the medication? It's important to note that in any trial, you're going to have a significant placebo effect. And this was even seen in a similar neurodevelopmental disorder called Rett syndrome. And there was a phase 3 placebo-controlled and blinded trial for a drug called Rett, which subsequently was approved by the FDA. And there was a particular measure called the Rett Syndrome Behavior Questionnaire. And this is the change in that score on this axis versus different time points along the study along the x-axis here. But as you can see, the people that got placebo, which is the dotted line here, appeared after week 2, and even at week 6 were performing in a similar fashion compared to the people that were actually getting drug. And it was only at the 12-week mark that there was a significant difference between those two groups. So therefore, it's important to think about having placebo and blinded controls in trials in order to best understand whether or not something's truly helping. So future trials in GRIN disorders, I think, are going to depend on the prior knowledge of the functional status as to whether or not you gain of function or loss of function. There have been trials in this to this point. They've all been non-blinded and non-randomized. For GRIN 2A and GRIN 2B, basically what you're looking for would be improvements in behavior, development, EEG, and seizures. And so the trial from Johannes's group, published by lead author by Dr. Cray in 2022, there were 9 folks in that trial, and it's important to note that one of those people got worse, and it turned out that they were gain of function. Then there's two trials that came out of the group in Spain, and the largest was just recently published with 24 individuals, and it was non-blinded and non-randomized. And it's important to note that these trials suggested that L-serine was safe, but it wasn't clear whether or not that there were enough people to really show that there was truly a benefit or not. There have been a number of trials with individuals with gain of function changes, looking at drugs like memantine, dextromethorphan, magnesium, and ketamine. And again, these were looking for very sick individuals who had medically refractory epilepsy and status epilepticus. And these are the different studies that have been done there. Again, it's a relatively small number of individuals. So my opinions, these are very helpful to understand the safety and support going forward with standard clinical trials. And the larger trials are really needed to understand who's going to respond and how much and what is safe. And I think that N-of-1 should be only for extreme situations where there's ICU or other similar serious hospital admissions, multiple daily seizures, et cetera, and when the functional status is known. And your clinicians can contact me for recommendations about what a group of us have put together as to what might be a way of doing this. And so until a real trial comes along, I think there's some issues and suggestions for use of these drugs outside of a trial. I think everybody needs to understand your risk tolerance and understand that if you participate in one of these N-of-1 trials, it could prevent or delay your participation in another clinical trial, because most of these are going to say things like, well, if you've been on L-serine before, you can't participate in this trial, because it might potentially contaminate the results. Knowing your functional status is, of course, imperative. And you want to work with your clinical team to determine safety and efficacy criteria and understand many of these things that might be suggested may or may not be covered by insurance. Think about what blood work or other studies to be done, such as a heart test. Should EEGs be done, Vineland's, Bailey's, Mullins? How are you going to know if what you're doing is really helping? And then, of course, how often are you going to repeat these studies? And of course, you can have your clinical team contact us for suggestions. And there is the possibility for you to enter your data into the registry before and during treatment so that it could potentially be tracked. Otherwise, all of these studies are not going to be captured, and nobody's going to be able to know, how this could be useful to the broader community. So we are, because of a lot of interest in this from the community, we're going to do our best to try to put together a clinical trial. And I want you to keep your fingers crossed that we're successful in funding. And we're going to approach a mechanism called NeuroNext, which is part of the National Institutes of Neurologic Disorder and Stroke. There's an infrastructure grant, and we can apply to this for a clinical trial. One of the important things that we have to have in place is something called an investigational new drug approval from the FDA before we're even able to apply for the money from them. But I think the best way to approach this is with a double-blind, randomized, placebo-controlled trial where we follow people for 12 to 24 weeks. I think it would be good to have 50 at a minimum. 100 individuals would be great. For L-serine, we would want to look at likely or probable loss of function or nonsense variants. And if somebody has a variant that's not known, we want to be able to test this through CFIRB and Dr. Trainellis's lab. I think it's important as part of the trial, we'd like to do a dose escalation to a maximum tolerated dose to, again, find out what's the right dose for people. And of course, we want to do a lot of laboratory studies and follow very closely everything that happens to you during the trial. Even if unfortunately you break your arm, we still need to keep track of that because that's the sort of thing that the FDA and all studies would want to know as part of a clinical trial. Anything that happens to you. We need to define what our efficacy measure is. Efficacy measure is going to be, I think it's probably going to be things like seizures, diaries, and then other developmental measures such as Vineland's, Bailey's, maybe a clinical severity assessment such as a CCSA, probably EEGs, and then probably, because the FDA likes it, something called a clinical global impression of improvement. With the NeuroNext mechanism, we can do sites in both North America, US, and Europe. So I think everybody should be able to participate, which I think is really exciting about this mechanism. So thank you very much for your attention. Thank you very much to all of the families that have participated in the registry. Please reach out if you have any questions. And thanks very much to the funders of the registry, Citizens United for Research in Epilepsy, the GRIN2B Foundation, and the Simons Foundation. Thanks again.
Everyone, thank you so much for your time and attention. Let's see if there are any questions in the chat that for our current speakers that might be able to be answered. We only have about a minute left, and then we're going to transition to our next session. It looks like Dr. Chung addressed a lot of the questions that were put in the chat earlier. Let's see. There was one question about what happens when a variant is indeterminate, gain of function, loss of function. Does anybody want to take a stab at that?
I guess it becomes indeterminate because it hasn't been tested before. So the web page sort of tries to guess. And Tim explained in the end that they will test it down the way. So for the GRINs, they will test it. For the GRIA, we will test it. So we will get there. Sometimes it just takes a little bit longer than expected.
Sounds good. One more question, and then we'll transition. So again, we're all staying in this session because we're going to be transitioning to our next keynote. So everybody stay put. But our next question is, can someone compare and contrast the databases, registries, and surveys? They say that it's difficult to know where to focus. So any thoughts or feedback on that?
So we are working closely together across the different databases, but there will be some overlap. But clearly, if you have a GRIN disorder, you could choose to contact Simons, or you can choose to contact Tim or Johannes, depending on where you live, you don't need to put your data in hours. It would be better, more appropriate to use those. If you have a GRI disorder, you can choose hours. So, but I mean, we still do collaborate closely and do share data anonymized. So I think in the end we will get there, but we don't expect the families to put their data in all the registries. You can also cherry pick, but I think it's important that you get involved.
I agree. It's important to get involved and be counted for everything in Simon's Searchlight. Just to be clear, I'm not actually a researcher using the data. I'm really here to be able to make sure the data can be used by anyone and everyone in a responsible way. So that's a lot of what we do with the Searchlight program.
Wonderful. Thank you both so much for your feedback. Thanks to everybody for your time and attention. Thank you, Dr. Byatt, for being on call and taking the time to be with us today. And thank you so much, Dr. Chung, and to Dr. Benke, who couldn't join us live today, but we're very grateful for all of your insights related to the registries and the phenotypes for GRI disorders. It was a very informative session. So now we are going to stay here. Again, thank you again for your participation. This session was sponsored by GRIN Therapeutics, and we greatly appreciate their support.